What keeps life going, generation after generation, even as every individual organism faces certain death? The answer lies in a process so fundamental, yet often overlooked. In this journey, we'll uncover how reproduction ensures the continuity of life, exploring the mechanisms that allow species to persist and adapt across millennia. After our opening question, let's preview the journey ahead. We'll explore the types of reproduction, the stages of life, the intricate events of sexual reproduction, and see how these processes unfold in both plants and animals. Reproduction comes in two main forms: asexual and sexual. Each mode shapes the fate of offspring and the survival of species in unique ways. Let's dive into how these processes work and why they matter. Before we go further, let's clarify a key term. In biology, a clone is a group of morphologically and genetically similar individuals produced by asexual reproduction. This concept is central to understanding how asexual reproduction works and why its offspring are so similar. Asexual reproduction takes several forms, each with its own mechanism. Let's look at the main ways organisms reproduce without the need for two parents. Binary fission is a process where a cell divides into two halves, as seen in organisms like amoeba and paramecium. Budding involves an unequal division that forms buds, which is common in yeast and hydra. Vegetative propagation allows new plants to grow from nodes, rhizomes, or tubers, as seen in potatoes and ginger. Asexual structures like zoospores, canidia, and gemmules are found in algae, fungi, and sponges. Sexual reproduction is more complex, involving the formation and fusion of gametes. This process introduces genetic diversity, setting the stage for adaptation and evolution. Gamete formation involves producing male and female gametes, such as sperm and egg. Fusion of gametes, known as syngamy, results in the formation of a zygote through fertilization. Genetic variation means that offspring are not identical to their parents, which is a hallmark of sexual reproduction. Let's weigh the differences between asexual and sexual reproduction. Each mode offers distinct advantages and challenges for organisms. Asexual reproduction relies on a single parent, producing offspring that are clones. It's a rapid and straightforward process, often seen in organisms with simple structures. Sexual reproduction requires two parents and results in offspring with genetic variation. Though more complex and slower, it's the preferred mode in higher organisms. Every organism has a unique lifespan, and its journey from birth to death is marked by distinct phases. Let's explore how these stages shape reproductive timing and strategies. Lifespan is a fascinating concept, revealing how organisms manage survival and continuity. Let's break down the key insights. The period from birth to death, known as the lifespan, can range from just a few days to several thousand years, depending on the organism. Single-celled organisms are unique in that they do not experience natural death, unlike multicellular organisms. Species continuity is maintained through reproduction, allowing life to persist across generations. Organisms transition through distinct phases: juvenile, reproductive, and senescent, each shaping their reproductive strategies and timing. The juvenile phase is the period of growth before an organism reaches reproductive maturity. The reproductive phase is when organisms actively reproduce and form gametes. The senescent phase marks old age, with a decline in reproductive activity and eventual death. This question challenges our assumptions about plant life cycles. Why do some species, like bamboo, flower just once after decades, while others bloom annually? The answer lies in the intricate interplay between genetics, environment, and evolutionary strategy. By exploring this, we gain insight into how reproductive phases are adapted for survival. Reproductive timing varies widely. Some animals breed only in certain seasons, while others are continuously active. This diversity reflects adaptation to environmental conditions. Seasonal breeders, such as birds and wild mammals, reproduce only during favorable seasons, ensuring the best chances for offspring survival. Continuous breeders remain reproductively active throughout their reproductive phase, as seen in many mammals. Sexual reproduction unfolds through a series of well-defined events. From the creation of gametes to the development of a new organism, each step is crucial. Let's trace this journey. Here's how the process of sexual reproduction unfolds, step by step. Each stage builds on the previous, leading to the creation of new life. Gametogenesis is the first step, where male and female gametes are formed. Gamete transfer brings the male and female gametes together, setting the stage for fertilization. Fertilization, or syngamy, is the fusion of gametes, resulting in a diploid zygote. Zygote formation marks the creation of a new cell, the starting point for embryogenesis. Embryogenesis is the final stage, where the zygote develops into an embryo, eventually becoming a new organism. Gametes come in different forms. Some are nearly identical, while others are clearly distinct. This diversity shapes the reproductive strategies of various organisms. Isogametes are morphologically similar gametes, as seen in Cladophora. Heterogametes are distinct male and female gametes, found in organisms like fucus and humans. Notice how chromosome numbers differ between myocytes and gametes across species. This comparison reveals the importance of reduction division in sexual reproduction. Humans have 46 chromosomes in myocytes and 23 in gametes, highlighting the reduction division. Houseflies show 12 chromosomes in myocytes, with gamete numbers not specified. Rats have 21 chromosomes in gametes, with myocyte numbers not specified. Dogs have 78 chromosomes in myocytes, with gamete numbers not specified. Cats have 19 chromosomes in gametes, with myocyte numbers not specified. Fruit flies have 8 chromosomes in myocytes, with gamete numbers not specified. Ophioglossum, a fern, has 630 chromosomes in gametes, an unusually high number. Apples have 34 chromosomes in myocytes, with gamete numbers not specified. Rice has 12 chromosomes in gametes, with myocyte numbers not specified. Maize has 20 chromosomes in myocytes, with gamete numbers not specified. Potatoes have 24 chromosomes in gametes, with myocyte numbers not specified. Butterflies have 380 chromosomes in myocytes, with gamete numbers not specified. Onions have 16 chromosomes in gametes, with myocyte numbers not specified. Fertilization can occur externally or internally, and in some cases, organisms bypass fertilization altogether. These variations reflect the adaptability of reproductive strategies. External fertilization is when syngamy occurs outside the body, as seen in algae, fishes, and amphibians. Internal fertilization happens inside the body, typical of reptiles, mammals, and many plants. Parthenogenesis is the development of new organisms without fertilization, observed in rotifers, honeybees, and some lizards and birds. After fertilization, the zygote develops into an embryo. The method of development, whether outside or inside the body, has profound effects on offspring survival. Oviparous animals lay eggs outside the body, as seen in reptiles and birds. Viviparous animals have young that develop inside the body, typical of mammals and humans. Embryonic care is more advanced in viviparous organisms, leading to higher survival rates for offspring. To bring these concepts to life, let's examine how reproduction unfolds in plants and animals. Their strategies reveal the diversity and ingenuity of nature. Plants use a variety of vegetative propagation methods to reproduce. These strategies allow for rapid and efficient multiplication, often exploited in agriculture. Runners are horizontal stems that produce new plants, commonly seen in grasses. Rhizomes are underground stems, as found in ginger and banana, that give rise to new plants. Tubers are swollen underground stems, like those in potatoes, which sprout new plants. Bulbils are small bulb-like structures, such as those in agave, that can grow into new plants. Leaf buds form on the margins of leaves, as seen in bryophyllum, and can develop into new plants. Sexuality varies widely in plants and animals. Some species combine both reproductive organs, while others keep them separate. This diversity shapes their reproductive strategies. Bisexual plants, like chara and sweet potato, have both male and female reproductive structures. Unisexual plants, such as marchantia and papaya, have separate male and female individuals. Hermaphrodites are animals like earthworm, sponge, and leech that possess both male and female reproductive organs. Unisexual animals, such as cockroach, have distinct male and female individuals. Pollination is a critical step in the sexual reproduction of flowering plants. The method of pollen transfer can determine the genetic diversity and success of the next generation. Self-pollination occurs when pollen is transferred within the same flower, as seen in peas. Cross-pollination involves transferring pollen between different flowers, typical of dioecious plants. Pollination mechanisms are specialized events that ensure successful transfer of pollen grains. After fertilization, plants undergo embryogenesis, leading to the formation of seeds and fruits. This process ensures the propagation and protection of the next generation. Zygote formation takes place inside the ovule, marking the start of a new plant generation. Seed development follows fertilization, with ovules maturing into seeds. Fruit formation happens as the ovary develops into a protective structure for seeds. The story of reproduction is the story of life's resilience. From simple clones to complex offspring, every strategy ensures survival. Through a dazzling array of reproductive mechanisms, nature adapts, evolves, and persists. The diversity of reproduction is not just a biological fact. It's the foundation of life's continuity.